On this trip through the digestive system, we will study digestion, the enzymatic breakdown of food to components that can be absorbed, and absorption, the movement of nutrients, salts, and water across the GI epithelium into blood or lymph. Your goals for learning are to describe the action of the digestive enzymes and bile salts, to describe transepithelial transport of monosaccharides and amino acids, to explain the role of bile salts as emulsifiers essential to digestion and absorption of fat. Here's what you need to know: the chemical structure of major nutrients, how pH affects enzyme activity, how molecules diffuse across membranes, how carriers transport molecules across membranes. To see definitions of terms, click the bold red words. Click the food to begin. Ingested food is first broken down mechanically into pieces that can be swallowed, then into small particles that can pass out of the stomach into the small intestine. Click the small particles. Small particles contain the major nutrients in our diet: carbohydrates, protein, and fat. Enzymes digest small particles into components that can be absorbed. In this topic, we will study the enzymes that digest each of these major nutrients. Click carbohydrates to study their digestion. The most abundant dietary carbohydrates are starch and the 3 disaccharides: sucrose, or table sugar, lactose, or milk sugar, and maltose, or grain sugar. Carbohydrates are built from monosaccharides and must be digested to their component monosaccharides so they can be absorbed. In our representative molecules, geometric symbols represent single monosaccharides. Starch is digested to glucose. Sucrose is digested to glucose and fructose. Lactose is digested to glucose and galactose. Maltose is digested to glucose. Click protein to study its digestion. Proteins are digested to amino acids and small peptide chains of 2 or 3 amino acids. In our representative protein, each circle represents a single amino acid. Click fat to study its digestion. Most dietary fat consists of triglycerides. Triglycerides are digested to monoglycerides and free fatty acids. In our representative triglyceride, the 3 circles represent the carbons of the glycerol backbone, and the picket fences represent fatty acids. Remember that nonpolar products of digestion can be absorbed across the intestinal epithelium by simple diffusion, a passive process. In general, polar substances are absorbed by carrier-mediated transport. Carrier-mediated transport may be active, requiring the energy of ATP, or passive, relying on the concentration gradient of the substance being absorbed. Plant starch and glycogen, animal starch, are the most abundant dietary carbohydrates. Carbohydrates are long polymers of glucose. Digestion of starch begins in the mouth with salivary amylase, which is maximally active at about pH 7. Drag starch to amylase to begin. Amylase breaks starch down to the disaccharide maltose, to fragments of 3 glucose molecules called maltotriose, and to small branched fragments called limit dextrins. In our illustration, each hexagon represents a glucose molecule. The hexagons at branch points represent glucose-glucose links that are not hydrolyzed by amylase. Click a digestion product to continue. Amylase continues to work in the stomach until food is mixed with gastric juice and acidified. Acid denatures amylase. In the stomach, pepsin begins the digestion of protein. Proteins are long polymers of amino acids. Pepsin is the only major digestive enzyme that is maximally active at acidic pH. Drag the protein to pepsin to begin. Pepsin cleaves proteins at bonds between the amino acids tyrosine and phenylalanine, producing peptide fragments and a small amount of individual amino acids. In this illustration, each circle represents an amino acid. Click a peptide to continue. Pepsin is active only in the stomach. When chyme is neutralized in the duodenum, pepsin is denatured. Most digestion and almost all absorption occurs in the small intestine. The pancreas supplies the digestive enzymes for most foodstuffs. Neither salivary amylase nor pepsin is essential for digestion. Pancreatic enzymes alone are adequate to digest carbohydrates and protein. There is little digestion of fat until it reaches the small intestine. Pancreatic lipase is essential for fat digestion. Without it, malabsorption of fat occurs. Humans lack digestive enzymes for the plant polysaccharide cellulose and some other complex carbohydrates. Drag a vegetable to the intestines. Vegetables and fruits are good sources of vitamins, minerals, and other nutrients. The indigestible polysaccharides contained in vegetables and fruits make up dietary fiber. Fiber increases bulk of the stool and promotes its timely movement through the colon. Click the intestine to observe salt and water absorption. Most of the salt and water that enter the GI tract are absorbed in the small intestine. Salt and water may be absorbed by transcellular transport through intestinal epithelial cells or by paracellular transport through leaky tight junctions between cells. Paracellular transport is passive and keeps the total osmolarity of the intestinal contents similar to the osmolarity of blood plasma. Transcellular transport of fluid depends on active transport of sodium. Sodium-potassium pumps in the basolateral membrane pump sodium out of intestinal epithelial cells to keep the intracellular concentration of sodium low and to elevate the concentration of sodium in the interstitial fluid. Sodium enters epithelial cells at the luminal surface, co-transported with glucose or amino acids through sodium ion channels and on several other transporters, moving down its electrochemical gradient. Basically, chloride follows sodium and the osmotic pressure generated by the movements of salt and other solutes into the interstitial space causes water to follow by osmosis. These transport mechanisms are similar to salt and water transport mechanisms in the proximal tubule of the kidney and may be reviewed in the urinary system module. The small intestine has the capacity to absorb more food than is usually ingested at one time. Increased intake causes increased absorptive capacity. It is almost impossible to exceed the absorptive capacity of the small intestine. We will study absorption of the end products of digestion of the major nutrients on the next pages. The carbohydrates that enter the small intestine for digestion include starch, its breakdown products from salivary amylase digestion, and the dietary disaccharides, sucrose, table sugar, lactose, milk sugar, and maltose, grain sugar. Drag starch to amylase to begin. Pancreatic amylase continues the breakdown of starch in the lumen. It is identical to salivary amylase and maximally active at about pH 7. Since only monosaccharides are absorbed, the breakdown products of starch and the disaccharides must be digested further. Brush border enzymes accomplish this job. Click an epithelial cell to continue. A brush border enzyme called glucoamylase that lies close to the sodium glucose cotransporter breaks down maltose and maltotriose. Drag maltotriose to its enzyme. A brush border enzyme called dextrinase breaks down small branched segments of starch. It also lies close to the sodium glucose cotransporter. Drag limit dextrin to its enzyme. Brush border enzymes also break down sucrose and lactose. Click the brush border membrane. Use what you know about enzyme names to drag sucrose and lactose to their respective enzymes. Drag glucose to a transporter. Most nutrients are absorbed by transepithelial transport, moving first into the intestinal epithelial cells at their luminal surface, then out at their basal surface. Glucose and galactose enter intestinal cells, co-transported with sodium by the process of secondary active transport. Notice that both glucose and galactose use the same transporter. Drag fructose to its transporter. Fructose enters intestinal cells on a fructose-specific transporter by the process of facilitated diffusion. Click the membrane to continue. All monosaccharides leave epithelial cells on a common transporter by the process of facilitated diffusion. Drag a monosaccharide to the transporter. Notice that glucose, galactose, and fructose use the same transporter. They enter capillaries for transport in the blood. Ingested proteins, peptides from pepsin digestion, proteins from sloughed cells, and enzymes enter the small intestine for digestion. Trypsin, chymotrypsin, and carboxypeptidase, the major pancreatic proteases, continue protein digestion in the lumen. These enzymes are maximally active at about pH 7. Drag a protein to trypsin to begin. Trypsin and chymotrypsin cleave proteins into smaller peptides and some single amino acids. Carboxypeptidase cleaves one amino acid at a time from the carboxyl end of a protein. Dipeptides and tripeptides as well as single amino acids are absorbed. Click an epithelial cell to continue. Aminopeptidase and dipeptidase continue the digestion of peptides. Drag a peptide to aminopeptidase. Click the membrane to continue. Many single amino acids enter intestinal cells co-transported with sodium by the process of secondary active transport. There are several different amino acid transporters. Transport of some amino acids does not require sodium. Most proteins absorbed as di- and tripeptides. Small peptide co-transporters use secondary active transport, some driven by sodium, others by hydrogen ions. Click a tripeptide inside the cell. Di- and tripeptides are broken down to amino acids inside the cells. Click the basolateral membrane to continue. Some amino acids leave epithelial cells by diffusion. The more hydrophobic the amino acid, the more likely it is to lead by diffusion. Other amino acids leave cells by facilitated diffusion or co-transport with sodium. Triglycerides are the most abundant dietary fat. They are made from a glycerol molecule to which 3 fatty acids are attached. Fatty acids may be short, medium, or long chains of carbon and hydrogen. Almost all fat digestion occurs in the small intestine. Pancreatic lipase breaks down triglycerides. It is maximally active at about pH 7. Since fat is insoluble in water, it tends to aggregate in large drops, leaving little surface area for lipase to work. Segmentation in the small intestine breaks the large drops into smaller drops and disperses those drops throughout the chyme. Click the large fat globule. Segmentation in the small intestine does to chyme what shaking does to a bottle of oil and vinegar salad dressing. It disperses the fat layer into the aqueous layer. Notice what happens when you set the jar down after shaking it. Click the bottle to continue. Bile salts in the intestine keep the small fat droplets in solution by a process called emulsification. The function of bile salts is directly related to their structure. They are hydrophobic on one side, their steroid core, and hydrophilic on the other side, primarily due to the presence of hydroxyl groups. Drag bile salts to the smaller drops. Keeping the small fat droplets in solution provides a large surface area for lipase action. Drag lipase to the small droplet. Lipase digests triglycerides to monoglycerides and free fatty acids. Drag a bile salt to the breakdown product. Another function of bile salts is to surround the cleaved products, forming tiny droplets called micelles. Micelles are 1,000,000 times smaller than emulsified fat droplets. When micelles are in close proximity to the cell membrane, monoglycerides and fatty acids move out of them to enter intestinal cells by simple diffusion through the lipid bilayer. Click a micelle. Triglycerides are reassembled inside the cells and packaged into chylomicrons that are coated with lipoproteins to keep them emulsified. Drag a chylomicron to the basal surface of the cell. Chylomicrons leave the cell by exocytosis. Since they are too large to pass through the basement membrane of capillaries, they enter lymph vessels called lacteals. Click a fatty acid inside the intestinal cell. Short-chain and medium-chain fatty acids are absorbed by simple diffusion and can directly enter capillaries. However, the normal diet contains few fatty acids of these chain lengths. Cells and secretions of the large intestine have no digestive functions. Resident bacteria break down some of the indigestible carbohydrates and use the released nutrients. These bacteria synthesize some B-complex vitamins and significant amounts of vitamin K that are absorbed. Vitamin K is essential for liver synthesis of some blood-clotting proteins. The colon absorbs small amounts of salt and water as feces pass through it. That completes our third and final trip through the GI tract. Here's a summary of what we've covered. Digestion occurs primarily in the small intestine by action of pancreatic enzymes. Absorption of most salt, water, and nutrients occurs in the small intestine. Most nutrients are absorbed by transepithelial transport. Monosaccharides and amino acids enter the bloodstream. Most fat enters the lymph. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.